eminent scientists. Today, I will be talking about inverse regulatory role of P53 modulating human sodium iodide symporter gene expression in breast cancer. So one of the focus of our group is we work on human sodium iodide symporter. To start with, human sodium iodide symporter, also known as HNIS, is an 643 amino acid containing intrinsic plasma membrane glycoprotein. It is also known as SLC5A5 as it is a member of solute carrier transporter family. It is majorly expressed in basolateral cells of the thyroid follicular cells, and as the name indicates, it co-transports two sodium ions along with the one iodide ion using energy gradient generated by sodium-potassium ATPase. Once inside the cell, it is used for organification and synthesis of thyroid hormones. This is the putative structure of the protein showing 13 transmembrane LLCs, and the ability of this protein to accumulate iodide is being exploited since many years in the dual imaging for the well-differentiated thyroid cancers. Now, apart from thyroid, this protein is also expressed in salivary gland, gastric mucosa, small intestine, and lactating mammary gland. Now, why this protein is so important to study? In a seminal study by Taziba et al., which was published in Nature in 2000, it was shown for the first time the aberrant expression of this protein in breast malignancies over the normal breast tissue. This protein is not expressed in the normal breast tissue. It gets upregulated during lactation to provide newborn the higher iodine for the synthesis of the hormones. But its aberrant expression was observed in breast malignancies, indicating its potential role in breast cancer management. Thereafter, there are numerous studies showing the functional uptake of the technetium pertectinate to this protein, and not only the primary breast tumors, but also the breast cancer metastatic lesions also show the expression of this protein. A couple of years ago, our own group conducted a thorough analysis of intensity-specific staining of this protein in different breast cancer subtypes. This is the first study in which different breast cancer subtype-specific intensity was studied. This is a representative image showing the NIS antibody stained tumor tissue sections. And what we found in this is the y-axis represents the number of the patients, x-axis represents the different breast cancer subtypes, while the z-axis represents the intensity of the staining. So more than 70% of the patients show positive staining for this protein, and if the intensity is considered, in this group, around 30% of the patients have very intense staining for this protein, which is compared to that of the thyroid gland, for which NIS-based radioiodide therapy is already being used in clinics. So all these studies project the role of NIS in breast cancer management. It is an endogenous protein, so it is non-immunogenic. Immunogenicity is the major hurdle, as we all know in gene therapy trials. And this protein, tissue distribution is quite limited, and it can be used for dual imaging using various radioisotopes like iodine 123, 124, and also the higher energy isotopes like iodine 131 and rhenium. But there are few challenges. Insufficient expression of the functional protein for effective iodide uptake, and also low organification and retention of iodide in the breast tissue. So to overcome these challenges, the gene regulation of this protein in breast cancer should be thoroughly understood. What is known about its regulation in breast cancer is, it is very well known that NIS gene is differentially regulated in thyroid and breast cancer. But what are the putative regulators or the transcriptional regulators of its expression and cell signaling pathways involved in breast tissue are entirely unknown. In this cartoon, it is shown the well-known regulators and signaling pathways which are known to regulate NIS expression in thyroid, while retinoic acid is very well a known potent inducer of NIS expression in breast cancer, while the PI3K pathway and the JAK-STAT pathway are supposed to be involved in its regulation, but it's not known. MKX2.5, which is a homeobox containing transcription factor, is known to involve in lactation-induced and retinoic acid-induced NIS expression in breast cancer. But other than these two reports, the regulators of NIS expression in breast cancers are entirely unknown. So, To gain more insight on this issue, we aim to identify potential transcriptional regulators of this gene to achieve better therapeutic efficacy in breast cancer. We developed NIS promoter-driven biofusion sensor, which has FLUC2 as the bioluminescent protein and TurboFP as the fluorescent protein. This NIS promoter sequence was gifted from Dr. Hein, and the biofusion reporter was developed earlier in our lab. We know, I have done most of the studies in MCF7 cell line, which is a receptor positive cell line, but other than that, we have also included ZR75 and MDMB453. Out of these three cell lines represent two major breast cancer subtypes, and this is a representative image of MCF7 engineered cells showing the fluorescence of turboprotein. As I have told you, we have this promoter sequence with our hand. So initially, we have done in vitro analysis, in silico analysis in order to characterize the different transcription factor binding sites present on this promoter. This is a graphical output. We have used genomatics software, and what we have found is there are around 64 transcription factor binding sites present on the promoter. It includes thyroid-specific or thyroid-enriched transcription factors like PAX8, MKX2.1, PAX5, and also general transcription factors or ubiquitous transcription factors like CREB, AP1, RXR, MKX2.5, P53. Now, to making it more simpler and interesting, we have found 3 P53 binding sites present on the NIS promoter. This is the classical P53 conjunction sequence which shows RRR, CNNG, YYY separated by 0 to 13 bases. Again, RRR, CNNG, YYY. This represents the full P53 binding sites. It is generally well accepted that to the core CNNG, P53 binds as a dimer and regulates the expression of its downstream gene. On our promoter, we have found 3 sites, out of which 2 are full binding sites and 1 is the half binding site. The results of this analysis were also verified for the another software, that is bio-based transfact, and we have found similar findings. While analyzing for TF binding sites, we were also interested in generating the regulatory network or the transcription factors which are involved in the NIS gene regulation in breast cancer. This is the representation of the BKL pathfinder. What we found is, other than the known regulators of NIS expression in breast cancer like PAX8 and CREB, we have also found SP1. SP1 binds to the GC box on NIS promoter and it is required for its full functional activity. At the same time, we have found that SP1 is also regulating SLC5A5. Along with it, it also regulates TP53. And we have found binding sites of this on this promoter. So, is there any direct link between these two proteins? With this in mind, we ask the question, is NIS a direct transcriptional target of P53? For this, we have transfected our NIS promoter reporter sensor in 2 different cell lines, MCF7 and MDMB453. And the effect on the promoter activity was checked after the activation of endogenous P53 by doxorubicin treatment. DR indicates the doxorubicin treatment, and also the overexpression of exogenous wild type P53 as it is represented by SM3. The normalization was done using PK-driven RNA plasmid, and what we found is, with the activation and overexpression of wild type P53, there is a significant decrease in the promoter activity, and the effect is seen to be specific, as in MDMB453 cells, non-significant changes in the promoter activity was found. But with the exogenous overexpression of wild type P53, we can see significant changes or significant downregulation in the promoter activity. The results are verified in another P53 wild type cell line, ZR75. This is the engineered cell line, stably expressing this promoter sensor, and we have found similar type of trend in this cell line as well. Further, when the promoter activity was checked after the transfection with P53 specific siRNA in MCF7 engineered cells stably expressing this sensor, we found significant increase in the promoter activity. So all this indicates activation and overexpression of P53 reduces NIS promoter activity and is effect found to be specific. Next, we validated these findings on the endogenous NIS level. First, we have checked its regulation at the transcript level using quantitative RT-PCR. Pacman gene chemistry was used, and the y-axis represents the relative mRNA levels. What we found is, with the treatment of the doxorubicin, there is significant reduction it was studied at the protein level using immunofluorescence study. The green indicates p53 specific staining, red indicates NIS specific staining, and blue indicates the nuclear DAPI staining. What we found is, after doxorubicin treatment, there is an activation and nuclear localization of p53. At the same time, it is resulting in the attenuation of the NIS staining. So, doxorubicin treatment activates wild type p53 and attenuates endogenous NIS expression. Further, these results are also validated in MCF7 NIS overexpressing cells. We have checked first at the transcript level, and we have found the similar trend as we have obtained in the parental cell line. When the effect was checked at the protein level using immunoblotting, in this immunoblot, 100 kD band shows fully mature glycosylated NIS protein, while the lower bands are the indicative of partially glycosylated NIS protein. We found that after the activation of wild type p53 and exogenous overexpression of p53, there is reduction in the NIS protein levels. Now, results at RNA and protein does not always guarantee the functional modulation. And as far as our protein is concerned and its potential therapeutic effect is concerned, we are very much interested to study is this is also reflecting at the functional level. And function of this protein is iodide uptake. So, this was tested in MCF7 cells, and we found similarly that with the activation and overexpression of wild type p53, it also significantly reduces NIS-mediated iodide uptake, showing the significance of the functional regulation. Now, in order to understand the regulation of this p53 of NIS in more details, we decided to proceed with two different approaches. First is to disrupt the p53 binding sites on NIS promoter and see what is its effect. And second is to see whether p53 is directly binding to the NIS promoter, exerting this modulation. First, we started with the mutagenesis, and we have taken the two strategies. First is AATT substitution. So, changing the core sequence CNNG to AATT, disrupting the site, and then checking the effect on promoter activity. And second is the site-specific deletion. So, delete the individual site one by one, and then check its effect on the promoter. Classical site-directed mutagenesis technique was followed. All the constructs were characterized by sequencing, and then they were used for transfection in MCF7 cells. The y-axis represents the fold change in the luciferase activity with compared to the full-length unmutated promoter. And what we found is mutation AATT substitution at site 3 and site 1 significantly increases NIS promoter activity, while with the mutation at site 2, we did not observe any significant changes. When the results were compared with that of the deletion set, we have found that similarly, especially the deletion at site 3 significantly upregulates NIS promoter activity. So both these results indicate the importance of site 3 and site 1 may be involved in p53 mediated regulation of NIS promoter. As I have said, we have parallelly we are also checking whether p53 is directly binding to the NIS promoter. We have used chromatin immunoprecipitation using p53 specific antibody, and this is the graph which when we have used the p53 binding site primer spanning the whole 3 sites. What we found, p53 binds to the NIS promoter, and there is a modest increase in its binding after doxorubicin treatment. Now, to narrow down further, and as from the suggestions from our SDM data, we have designed specific site 3 and site 1 specific primers and then check whether it is binding to these sites or not. So we found that p53 binds both site 3 and site 1, site 3 with more intensity, and its binding is increased further considerably after the doxorubicin treatment, especially at site 3, whereas the site 1 there is non-significant changes in the binding. These results will be verified further with the use of real-time PCR, but both these SDM and chip data indicate binding at site 3 is important for p53 mediated modulation of NIS promoter activity. We have found the repression of NIS by p53, and generally it is reported in the literature that half-binding sites of p53 are generally known to be repressive in their function. And if you remember, our site 3 is a half-binding site. So it is in agreement with that literature. What next? We want to validate our in vitro findings with the in vivo animal mouse model. Before proceeding for that, we have again verified the modulation of its doxorubicin with the live cell imaging, and as it is very clear, activated P53 decreases NIS promoter activity in dose-dependent manner in the live ZR75 cells. Then these cells are used. They were implanted orthotopically into mammary fat pad of the female biopsy nude mice. They were allowed to form tumors, and after the tumor formation, they were randomly divided into 3 groups: control group and the doxorubicin group, which received a single intraperitoneal injection of doxorubicin at the 2 different doses. We have selected the dose in such a way that it itself will not result in the tumor killing, but it will sufficient to induce the P53. This is the representative mouse images of each group, and what we found is, in the control group, there is bioluminescence signal continues to increase, but in the doxorubicin group, there is 40 to 50% reduction in the bioluminescence activity, bioluminescence signal. As this bioluminescence signal is a direct measure of NIS promoter activity, we can say that in vivo, these results are indicative of in vivo modulation of NIS promoter by P53. So to summarize all these findings, activated P53 represses NIS-mediated iodide uptake, and the P53 as a repressor of NIS function is reporting first time in the breast cancer, and we have checked its regulation at multiple levels, at RNA level, protein level, and at the functional level also. Our initial in vivo findings suggest the modulation of NIS promoter activity by activated P53. It will be carried out for in future with the large animal in each group. Along we wanted to see the effect of iodine 131 therapy in these in the in vivo model. And the take-home message I want to like to convey is, this is for the first time we are reporting the P53 mediated repression of NIS expression in breast cancer. As we all of know, P53 mutations are very common in cancer, and especially in breast cancer and in the triple negative breast cancer patients, which generally show very commonly the P53 mutations. They lack all the hormonal receptors, ER, PR and HER2. So the tumors are more aggressive and they are more challenging to treat. So in these patients, the triple negative patients with the mutant P53, and our IHC data also show moderate to high expression of NIS protein in these patients. So at least they are expected to have benefit from NIS-based radioiodine therapy in these patients. With this, I would like to acknowledge all my lab members, collaborators, Women Cancer Initiative funding for research support, ACTRAC for research fellowship and providing sponsorship to attend this meeting. This is our group and this is the institute where I work. Thank you.
Okay, any question for Ms. Keller? If I got the first question right. So you mentioned about the NIS expression, right, in the breast cancer. So what is the physiological function for this NIS in the breast cancer cell?
Okay. So in the normal situation, it is not expressed in the normal breast tissue. It is only seen in the lactation. And in the lactating phase, this protein is functional. It also uptakes iodide. And that iodide is used for the synthesis of thyroid hormone, as there is an increased need for the newborn for the synthesis of these thyroid hormones. In breast cancer, whether the protein is functional or not, there are 2 different schools of thoughts. One group of the literature have clearly shown that this protein is functionally present and it uptakes the radioisotopes like technetium or technetium. So using this function, can we target those breast cancer cells with the radioiodine 131 so to make it therapeutically beneficial? That is what our goal is.
Yeah, okay, thank you. Any other questions? Yes, please.
Very nice study, very impressive. I was just wondering, because thyroid cancer, the NIS has shown a very much effective efficacy on the radioiodide therapy. So is this the similar findings have been done with the P53 and the angle, P53 and NIS angle has been reported in the thyroid cancer?
No, it is not reported. How the P53 regulates NIS function in thyroid, that is not known. It may be same or it may be entirely different, because as such, the gene regulation of this protein is tissue specific. What regulates its expression in thyroid? It does not regulate its expression in breast. But such reports are not there.
Yeah, because P53 is a guardian gene for the tumors. And I was also wondering, when you have actually represses the function of NIS. So that means in the cancer, it sees in the beneficial, in the benefit of host. That's why, because P53 is a guardian gene, and if it is repressing NIS,
It might be possible because P53 is a tumor suppressor, and we have found the aberrant expression of this protein in breast malignancies, but not in the normal breast tissue. So it could be possible, but as we all of know that mutations are also very common. So at least in those mutant patients, we are expected to have higher risk, and our IC data also have indications that in TNBC samples we can expect moderate to high NIS levels. So at least they can be targeted with this radioiodine therapy using this protein.
one more question.
Sure. Go ahead.
So with your P53 binding sites, which you have seen, which binds to the NIS promoter here, so are they the mutant, they come under the mutant P53 or the wild type?
No, it is all done at the wild type level. We don't know at the mutant level.
Yeah, because that would be important if you are going for, in terms of the cancer, because cancer has the P53 mutations, and if that binding site is there, so then.
There are two approaches. This we are talking at the wild type protein and the promoter at the binding site. The second approach is in the mutant cell